So so the first thing that we're going to do, guys, is we are going to dig a pit, like onto the left hand side. That is going to be the inlet pit. What are we going to do? We are going to take all your waste material, like whether it is the waste from your animals, waste from your urine, your waste from your fecal matter, waste from your agriculture, waste from your household, whatever waste that you can get your hands on, that is going to be added onto the left hand side. That is going to be your inlet. Okay, now when you mix all these substances into it, guys, what do we call it? We call this thing as your slurry. Okay, now that slurry is slowly going to be deposited, or it is going to be put into your into your digester. Okay, the middle portion that is there, that is going to be digester. Now as it goes into the digester, guys, now we are going to see that, oh my god, okay, all the bacteria which are present out over there, okay, whether it is the anaerobic bacteria or whether it is the methanogens, okay, how it is going to happen, we are going to talk about it today. Okay, all these bacteria, they are now going to start acting onto it. Okay, and now they are going to break down all the polymers, and they are finally going to convert it into gases, like it is going to be your CH4, that is your methane. It is going to be your carbon dioxide. It is also going to be your H2S. It is going to be your H2, that is hydrogen, and other trace gases. Okay, now, okay, then what is going to happen, guys, whatever the spent thing is, like all that entire material, whatever that we are like spending out over here, that ultimately is now going to go on the other side across the separator. This middle line is called as a separator, and then slowly it starts coming out through the other side, which is called as the outlet. But now this thing, the one that is going to be coming out from the other side, guys, that is going to be the spent one, meaning it has already been used for your this formation, that is your biogas formation. So what do we call it, guys? We call this thing as your sludge. And remember, what is the most important thing regarding the sludge? That this sludge can be directly used as a source of fertilizer for the plant, because it has all the important materials present inside of it. So basically, this is how it is going to be. Now, I told you that, in this digester, we are going to have this dome. Now, what is this dome? This dome keeps on moving up and down depending upon the amount of gases present inside of it. If for any reason you are going to have high amounts of gas, then this dome rises onto itself. And then you are going to see that we have this wall. We can adjust the outlet out over here. And from there, the gas is going to go into the pipeline, and from the pipeline into your houses, for either your cooking purposes or for your lighting purposes or for your heating purposes. So that is what is going to happen out over here, guys. Okay, so this was that entire thing that we had talked about. Okay, but now when we talk about this thing, 1 minute. Okay, when we talk about this entire biogas plant, okay, so which are the biogas plants that are going to be generally used, and how does it really happen? Okay, so we have, like, two models out over here. Okay, generally the most important ones, like, see, I told you, like, in your textbook they have shown you these two things out over here. So I have drawn this. This is the most common model, and it is a different, a little bit variety. Okay, so in this model there are actually two models which are present, and these models, one is your KVIC, okay, and the second one is going to be your IARI, Indian Agricultural Research Institute. Okay, so that one. So these are the two ones that are there. Okay, but now the next important thing is, guys, what are the different steps that are going to happen out over here? Okay, the steps involved in your biogas formation. Meaning, see, we have to prepare this CH4, because what did we say, guys? What is biogas? Biogas is mainly going to be your methane. Okay, so biogas is nothing but it is methane. So first thing, guys, okay, the first step that we are going to have, 1 minute, has anybody messaged? No, no. Okay. Okay. Chalo. So let's try to see what are the what are the reactions that are going to happen out over here. Okay. Now remember, whatever that is happening out over here, that is nothing but your anaerobic digestion. Okay. So this is going to be anaerobic digestion. And this entire anaerobic digestion, it is going to include three important steps. Okay, so what are the first steps, guys? The first step, it is known as your hydrolysis. Okay, hydrolysis, or we also call this thing as solubilization. Okay, so hydrolysis or solubilization. Okay, so what is going to happen out over here? See here, guys, what is going to happen is you are going to have larger molecules. Now, when you're going to use your fecal matter, when you're going to use your, the waste from your household, like kitchen waste, or whether it is waste from your agricultural, okay, so like whether it is your peels of the vegetables, or it is going to be leftover parts of the vegetables, or leftover parts of your food, these are going to have large big amounts of your substances. So they are going to be polymers inside of it. Okay, so now what happens is, guys, all these polymers, okay, I need to convert it into monomers first. I cannot like directly work on these polymers. So you are going to see, guys, that we are going to have the polymers, and these polymers now, they are going to be first converted into the monomers. And how is this going to happen? This is going to happen in the presence of your anaerobic bacteria. Okay, this is the presence of anaerobic bacteria. But guys, the moment we are talking about anaerobic bacteria, who are we talking about? We are talking about two of the most important ones. One is Clostridium, and the second one is your Pseudomonas. Okay, Clostridium and Pseudomonas. These are going to be the anaerobic bacteria. Okay, fine. So now what is going to happen, guys? You are going to see that whatever we had added into this slurry, like the dung water, your the agricultural waste and all, those larger molecules, the polymers, they are first converted into your monomers in the presence of your anaerobic bacteria. That is going to be your Clostridium and Pseudomonas. That's it. Okay, and see, guys, if we talk about your mm, monomers, what are the monomers basically? See, if we are talking about carbohydrates, then this is going to be your sugars, simple sugar. Either it is going to be your glucose, or either this is going to be your glucose, fructose, or it is going to be your galactose. So that way, if this is your protein, then this is going to be your amino acid. If this is going to be your fats, then this is going to be fatty acids and glycerol. Okay, so this is how it is going to be there. So if you want, I can write it down that way also, but I believe everybody is okay with this thing. Quickly tell me, yes, if you are understanding, or should I put it down, like what are polymers and what are monomers also? Quickly, 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 guys, anybody quickly reply. Okay, okay, fine, okay, okay. Okay, so guys, now once we have got these monomers, okay, now the next thing is I am going to convert this into acids. Okay, so the next step is going to be your acidogenesis, or the other name for this is your acetogenesis, acidogenesis or acetogenesis. So let's try to see how is this going to be. So acidogenesis or acetogenesis. Okay. Now, what is the name? See, guys, the basic name is acidogenesis. Meaning, what are you going to do? You are going to convert these monomers into your organic acids, right? So now you are going to see that here we are going to have these monomers. We have taken the monomers, and once again, in the presence of your acidogenic bacteria, okay, acidogenic bacteria or acetogenic bacteria. Okay, acidogenic bacteria. Now you're going to convert these monomers into organic acids. And especially when we talk about organic acid, guys, what are we talking about? We are talking about acetic acid. Okay, acetic acid. Mhm. So what is this? CH3COOH. This is going to be your acetic acid. Or sometimes we are going to convert it into your formic acid. Can anybody quickly tell me where do you see formic acid? Anyone? Absolutely right. Very good, Himanshu. Very nice answer. I loved it. Okay. Now remember, it is going to be seen in the bites of your ants. Okay. Ants are going to have formic acid, and you're also going to see it in your bee sting. Now, whatever that is present in ants, that is like, very little. Concentrate it like, hundreds of times, and that is going to be in the case of your bee sting. Okay. So very nice. Very nice answer, guys. Okay, so now From your monomers, now slowly we have converted it into your acids, that is either your acetic acid or formic acid. But now if I'm talking about this acidogenic bacteria, who are we talking about? We are talking about Acetobacter aceti. Okay, we have this Acetobacter aceti. Okay, because see, we are going to get acetic acid, so that has to be your Acetobacter aceti. Okay, but now guys, the next thing after this, the third step. Now after this, see, what do we have to remove? Guys, we need to remove your methane. Okay, I need to produce methane from here. So the third step that is going to happen, guys, that is nothing but methanogenesis. Okay, that is nothing but your methane formation. And now guys, what is going to happen? See, this organic acid, whatever that were there. Okay, now I'm going to ये क्या हो गया? Okay, so now guys, this organic acids, whatever that are there out over here, and especially I'm going to talk about your methanol, this thing as well, that is your acetic acid as well. Okay, so let's say this organic acids, we have this CH3COOH. Now this in the presence of your methanogenic bacteria now. In the presence of methanogenic bacteria, now what is going to happen? See guys, very, very simple. Okay, very simple stuff. Uh, One minute. No, 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 no. It is used to preserve dead bodies. No, 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 no. Formic acid is not used to preserve dead bodies. Formic acid, nahi, that is formalin or formaldehyde. That is different. Formic acid is different. Okay, don't confuse with formic acid and formaldehyde. Different stuff altogether. अरे, क्या हो गया? अरे वाह. Okay, ठीक है. Yeah, no problem, no problem. Let me just erase this, otherwise you'll get confused. अरे रे. Yeah, I just found out a new feature. See, guys, when I press the back button, it directly takes me to the last presentation that I was doing. And when I do the forward button, it takes me to the recent one. Yeah. Okay. So now guys, just look at this thing. Okay, so we have CH3COOH. Guys, what do we need? We want to produce your methane. So what are we going to see? See, guys, we have this. See, we have this carbon dioxide. I can just poof remove this carbon dioxide. And if I remove this carbon dioxide, guys, see what is going to remain behind. I am going to see CH4. CH4 is what? Methane plus it is going to be carbon dioxide. This is the simplest thing. Okay, but I'm going to give you something more. Okay, this is just to make you understand what exactly is going to happen. So you are going to see right out over here CH4 plus your CO2. Okay, this is the easiest way to understand, keep an idea about what is happening. But now, if we are talking about methanogenic bacteria, guys, who are we talking about? We are talking about methanobacillus. Okay, we have methanobacillus. Second, we have methanococcus, methanobacillus, methanococcus, and methanomonas. Guys, these are the bacteria which are going to bring about methanogenesis. Okay, methanobacillus, methanococcus, and methanomonas. Okay, but now let's see in reality what really is going to happen out over here, like how this thing really takes place. See, I told you the base reaction is going to be this thing. This is what we actually want. Okay, but now let's try to see how are these reactions going to happen one by one, one by one, one by one. So first thing, we are going to have 12 molecules. Okay, we start off with, we balance this thing and with 12 molecules. We have CH3COOH. This is going to give us, okay, in the presence of this methanogenic bacteria, okay, methanogenic bacteria, we are going to have 12CH4 plus 12CO2. This is the first reaction that is going to happen. Okay. Or the other way around. See, I told you we can also make use of this formic acid. So if we have to remove this formic acid, guys, then the situation is a little different. So in that case, we are going to have 4HCOOH. Okay. Now, guys, if I have to make CH4 from here, okay, so how will I get? So I if I see CH4, so I can have CH4 plus 3CO2 plus 2H2O. All of them coming from here only. Now let's see if it is balanced. See, I want 4C. So see, C is 1 out over here and this is 3C. So ho gaya 4C, 5. It is balanced. Okay, we have 4 and 2H. So 8H. So ये हो गया CH4. So 4 is out over here and 4 is out over here. And if that is the case, then rest everything is going to be balanced. Okay, everybody getting this thing, guys? Sure. Okay, and now the next thing, guys. Okay, next thing you're also going to see that whatever carbon dioxide that has been released. See, I told you carbon dioxide, and along with that, we are also going to release your H2O, right? So, sorry, your H2. So that H2, if I bind this thing together, so 4H2, guys, what are we going to get? We are going to get CH4 plus 2H2O, right? And all these things, guys, once again, what are they going to give us? They are going to give us our main thing, that is your CH4, CH4, CH4. And this is how we are going to get your methane. Everybody all right with this thing, guys? Sure, no problem. Okay, guys, once again, just have a look. Okay, just have a look at how these things have happened. Okay, so first of all, guys, we had larger molecules which are going to be your polymers. Slowly these polymers, they were broken down into monomers. Monomers were then converted into acids, and acids finally were broken down into your methane. Okay, just have a look. If you guys are okay, just see this thing. Then we'll move on to the next part, that is why should we go for this thing, that is why should we go for your biogas, and then we'll talk about your sewage treatment plants. Okay, everybody all right with this, guys? Can we move ahead? Yes, guys. Hello. Yes. Okay. Okay. Sure, let's move ahead, guys. Okay, sure. So guys, let's try to move ahead. Okay, and now let's try to see why are we going to go for this gobar gas. Okay, this gobar gas, basically biogas or gobar gas as we call it, it has many advantages over the normally used. Okay, the normally used, your whether it is your wood, or whether it is going to be your natural gas, or whether it is your coal. It has many advantages out over it. Let's try to see what are the ones. So the first thing, guys, the first and the foremost thing is this is a renewable source of energy, meaning this is inexhaustible. Rest others, whether it is wood, whether it is going to be your coal. Okay, these are all exhaustible sources. Okay, whether it is even your natural gas, it is an exhaustible source. Okay, so remember, This is a renewable source of energy. Others are exhaustible, non-renewable source of energy. Second thing, guys, this is going to be really, really cheap. Okay, it is going to be cheap, and second, this is going to be safe. Okay, cheap and safe meaning there is no harm to your entire life. Even if it catches fire, it is not going to burst out like your LPG, like a cylinder kind of thing. No, it doesn't happen that way. So very, very nice. So it is going to be cheap, it is going to be safe. Second thing it is going to be, guys, I mean the next thing it is going to be that this is going to be used for your industrial as well as domestic purposes. So it can be used for industrial and domestic purposes. So if I'm talking about domestic purposes, guys, what are we talking about? See, we are talking about lighting, like we are talking about your, lighting your lamps kind of thing. Okay, so first is lighting or street lighting. We can even use it for that purpose, guys. Okay, another thing is we can use this thing directly for cooking like we all normally do, or we can also use it for heating purposes, guys. So for all these things we can use this biogas. Okay, next thing, next important thing, guys, this is the one which is going to burn with a blue flame. Okay, so what is the difference between blue flame and yellow flame? Okay, now always remember, so the thing which burns with blue flame, we call this thing as your oxidizing flame. Okay, this is known as oxidizing flame. And the yellow one, that is going to be known as your reducing flame. Now, what happens is when you have that yellow flame, guys, you will always see that that yellow flame causes large amounts of pollution. It creates large amount of your gases. It creates large amounts of smoke. But with blue flame, guys, you are using LPG. LPG burns with a blue flame. After you cook your food, suddenly there is like, dhua, dhua, dhua in your kitchen. No, I don't think so. It is going to ever happen. Okay, so the reason is because this blue flame, it doesn't cause pollution. That is very, very important. So blue flame, it doesn't cause pollution, and plus this is going to be without any kind of smoke, like I said. Okay, however, the yellow one, that is a reducing flame, that is going to create loads and loads of smoke. Okay, but now the next thing, guys. Okay, the next thing we are going to have this. This is going to be your eco-friendly. This is going to be eco-friendly because it is not causing any kind of your pollution. Okay, next thing, this is also going to help in improving your sanitation. Because see, if you are going to prepare, okay, if you are going to prepare biogas, everybody is going to come and defecate into these, special toilets, okay, which are meant to be for your biogas. In fact, in your villages and all, people are given actually 2 rupees or 3 rupees to come and defecate into the biogas toilets. So it is a win-win situation. Okay, generally who pays for pooping? Nobody, right? But then what is this going to do? It is going to improve sanitation in the area. Why? Because then people will not defecate here and there. They will make sure that they will make use of these public urinals or public the latrines basically. So it improves sanitation. And lastly, but the most important thing, guys, okay, most important thing is this is going to be easily collected. Okay, this is going to be easily collected. It can be easily stored, and it can be easily transported from one place to another. Easily collected, easily stored, and easily transported. And oh, sorry, there's one more point. Okay, and guys, whatever the leftover part is, that the sludge, okay, that sludge, we can say that this can be directly used as fertilizer. Immediately this is directly used as fertilizer for your plants. So these are some of the important points that we need to know with regards to your this thing, your biogas. Okay, just have a look at this and then we'll start off with your sewage treatment plant, guys. Okay, chalo. So guys, let's try to move ahead. Okay, so now the next thing we are going to talk about is we are going to talk about your sewage treatment plant. Okay, now let's try to see how is this sewage treatment plant, guys. One of the most important questions. Okay, because this can be asked for two things. One, it is going to be asked for your drawing purposes, guys. And second thing, it is going to be asked for writing a short note. Now, earlier, when there was like, a 7 mark question, okay, so people never used to expect, a 7 mark question from, microbes and human welfare. But then, what used to be asked, guys, this sewage treatment plant. Draw a neat level diagram of sewage treatment plant and write a note on it, like, what really happens out over there. So that is very, very important question, guys. Now you are going to have maximum 4 marks. Okay, so this will be asked for 4 marks, guys. If they ever ask, wish to ask from this thing, then this will be asked for 4 marks, guys. Okay. Okay. So guys, now let's try to see what is going to happen in your entire biogas. It is going to be in your sewage treatment plant. So let's try to see this one by one, one by one, one by one. Okay. So first of all, guys, when we talk about this sewage, okay. So when we talk about sewage, what exactly do we mean by sewage? Sewage is nothing but all your waste waters. Okay. So like whatever waste that is coming from your bathroom, whatever waste is coming from your toilet, that is nothing but your sewage. Okay. So once again, when we talk about sewage, sewage is nothing but this is going to be your waste waters. Okay, and that is going to include your human waste that is inclusive of your fecal matter and your urine. Okay, both of them. So human waste, so if you want, I will just put down, this is going to be urine, this is going to be your fecal matter. Okay, but along with this, guys, what else can be used? Guys, you can use like different kinds of your this thing, that is your organic materials, any other kind of organic materials that you are using. So organic waste, any kind of those. And along with that, you can also use like your microbes in this thing, because these are all going to be a part of it. Now, where do microbes come into it? Guys, these are going to come from your fecal matter, because your intestines are going to be loaded with microorganisms. So whenever you are like defecating, along with the fecal matter, the microbes, they enter into your the sewage system. Okay, but now guys, see, there is going to be so much of water involved. See, whenever you go to the loo, guys, or whenever you go to the toilet, you are going to flush. But now, when you flush, have you ever really realized, like, how many like liters of water you are like flushing down the drain? See, for every flush, you're going to use around like 10 to 15 liters of water. That is how the tanks are going to be. Like, if it is a small tank, then minimum 10 liters. If it's a big tank, 15 liters. So every time you urinate, every time you defecate, 15 liters of water is like flushed down. Okay. But what happens to that flushed water? Are you going to reuse it? Like it goes to the septic tank in your building, then they are going to filter it, and once again they are going to use the same water, once again send it back into your bathrooms. No, 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 it doesn't happen that way. Then there has to be a huge process to remove that waste which are present inside. And for that reason, guys, we need to take this whatever sewage that we have produced, this needs to be taken to your sewage treatment plant. This is going to go into your sewage treatment plant. Okay, fine. Now, this sewage treatment plant, guys, we are going to call them as STPs, okay, sewage treatment plants. Now, what are these going to do? These sewage treatment plants, they are going to remove all your waste, okay, they remove, the sewage treatment plants removes waste, whatever that organic, inorganic matter, whatever the waste products that are there, and plus it is also going to remove your microbes. Okay, and now when it removes the waste from the water, it removes microbes from the water. Now you are going to get, like, your water. Now this is not going to be pure water, guys. This is going to be water which does not have microorganisms, does not have pollution as such. And this water can be now discarded. This, we call this thing as your effluent, basically. And this can be easily discarded into your water bodies. Okay, without this treatment, okay, we cannot like really discard the water into your water bodies, because what will happen is, if you just take this fecal matter water and you put it into some river or stream, immediately it is going to contaminate that thing also. However, on the contrary, in India, you will see many places where sewage water is like directly released into your water bodies. It is actually wrong. It is wrong. But then we can't help it, it is happening. Okay, so now there are like stricter regulations which are coming into place, where none of this water is actually going to be put into your streams or rivers or water bodies. It has to be treated. Okay, but now what is the sewage treatment plant going to be like, or what is this entire process? So STPs, basically, they are going to have four different kinds of things. Okay, so let's try to see. So the first thing, guys, you are going to see something that is called as preliminary treatment. Okay, so in preliminary treatment, see, guys, see, we don't really know what is going to be present in this thing, like in your sewage water. So we are going to try to remove, like, the coarse particles, the floating particles, whatever that are there, we try to remove in this thing, okay, in your preliminary treatment. In preliminary treatment also, we are going to have two treatments out over there. So we are going to have one, it is going to be screening. Okay, so this is going to be screening, and the second thing is, it is going to be your grit chamber. So screening and grit chamber. Okay, fine. Then after your preliminary treatment is done, now we move on to the second treatment. Okay, now this second treatment, this is going to be your actual treatment. So we call this thing as your primary treatment. Okay, so preliminary treatment, then we are going to have this primary treatment. Now, this primary treatment, guys, also remember, this is known as physical treatment. Why physical treatment? Because we are going to take care, or we are going to target the physical properties of the sewage, okay, sewage water. So this is also known as physical treatment. Okay, then the bracket. Okay, but now the next thing, guys, after your primary treatment, then we are going to go for your secondary treatment. Okay, so primary treatment, secondary treatment. Now in secondary treatment, what is going to happen, guys? You are going to make use of something that is, it is going to be like the biological treatment kind of thing. So you're going to target the biological properties out over here. So we say in the secondary treatment, this is your biological treatment or a biological process. Biological treatment, or this is going to be a biochemical process. Okay. And then, guys, after this, what are we going to have? After the secondary treatment, we are going to have the last treatment, that is tertiary treatment. This is going to be like your anaerobic digestion. Okay, so this is tertiary treatment. This is your anaerobic digestion. And here, what are we going to tackle? We are going to tackle both the properties. So this is going to be physical as well as biochemical treatment. Physical as well as biochemical treatment. Okay, so this is the overall map. Okay, this is the overall map of your sewage treatment plants. Okay with this thing? Sure, no issues, guys. Okay, so first we are going to go into your preliminary treatment, then we will slowly see what is going to happen out over there. Okay, so just have a look at how this thing works. Then we will slowly see, you know. Okay, chalo. So guys, let's try to see what exactly is going to happen in your preliminary treatment. Okay, and what is all this thing going to be like. So let's imagine we have collected, okay, we have collected all your sewage water. Okay, so your sewage water has entered. Now, first thing we are going to go for your screening. Okay, so let's say this is your sewage water. So this sewage water is first going to go, basically sewage. It is going to go into the first tank. Okay, so let's imagine this is one small tank out over here. Now, what is this tank, guys? This is nothing but your screening process. Okay, so in your screening process, so okay, so this is going to be screening. So now in your screening, guys, okay, now I am going to use large number of screens. Okay, so screens are what basically they are going to be different mesh sizes. Okay, and through these mesh sizes, what are you going to do? You are going to take the sewage and you're going to pass through the screens. Okay, so these screens basically they are going to be of different, okay, so different pore sizes, this thing, okay, or you call them as screens, or you can also call them as nets. Okay, so now what happens is, because you are like putting it now, either they can be horizontal or they can be vertical as well, depends upon what kind of you this thing you are using. So guys, when you are using this screening process, guys, what is going to happen, all the larger particles, okay, so all the suspended, okay, the major suspended particles, okay, or the floating materials, okay, the suspended particles or whatever that is floating, okay, or some coarse particles or your heavy particles, okay, so okay, so all these are going to be removed. Okay, so these are basically filtration, so these are going to be filtered and basically they are going to be removed. Okay, so this is the first thing that is going to happen in your screening process. Okay, fine. Now, some of this thing, okay, some of this basically, they are going to be larger particles, and also they can be easily filtered. Okay, fine. But now, once again, guys, once we have this water, now what are we going to do? We take this sewage water and now we are going to put it into the next chamber. So the next chamber, guys, we call this thing as our grit chamber. Okay, now why this is grit chamber, guys? The grit chamber, we call it because inside this, you are going to use various kinds of your stones, gravel, pebbles, okay, or you are going to have this entire brick ballast. Okay, so let's try to see what is going to happen. So the second thing, we are going to go into your grit chamber. Okay, so all this water, whatever that was there, so we have put it into the second thing. But in the second thing, we are going to have grids. So you are going to have these columns, how you see it in your water filter. There are these candles, and the candles are filled with some stony materials and all. Basically, so it is going to be that thing. Okay. Now, guys, so if I am having this stony material in your water filters also, is it really going to help in filtration process? The answer is yes. Guys, this is called as natural filtration, because the same thing, what is going to happen, whatever water that is going to be on the surface of the earth, it is going to slowly trickle down. And as it goes down, as it penetrates, guys, through the deeper layers of the soil, all the soil particles and whatever the contaminants are, they are going to be removed, and fresh water is going to go into the underground layers, and that is why we have the underground water, which is going to be fresh and it's going to be like pure kind of thing, because it has gone through that entire filtration through the earth's crust. Okay, so that is what is the idea basically. Okay, so now here we are going to have this grid chamber, and this grid chamber, what is this going to consist of, guys? So I told you this is going to have gravel. Okay, this is going to have pebbles. Okay, it is going to have small stones. Okay, okay, so it is going to have some small stones. And where is this? This is going to be in your brick ballast. Okay, now what is a brick ballast? Basically, this is nothing but reinforced with your bricks like this. So this is a brick ballast, right? So where else can you see a brick ballast, guys? If you see the traditional pizza ovens, okay, those are also nothing but brick ballast. Okay, so the Italian pizza ones, okay, so this is going to be seen in your brick ballast. Okay, but now, guys, what is going to happen because of this? So as this water is going to be there, guys, you are going to slowly see that, because of your gravity, because whenever you leave something like this, gravity is going to take care of it. So because of gravity, once again, guys, the coarse materials, whatever that are there, they are going to settle down and they get filtered into this grit chamber. Okay, so the coarse particles, these are going to be filtered by gravity. Okay. So guys, see, majority of the stuff, this is going to be removed. Okay. Now, once this thing is done, guys, now our next step. So whatever water that has been purified from here, now this is going to go for your primary treatment. Okay. But now, guys, what is this primary treatment? Primary treatment, we said it is going to be your physical treatment. Okay. So let's try to see. So once again, water from here, now it is going to go into your sedimentation tanks. Okay, so whatever we had done in the preliminary, okay, that was a little in a fast manner. Okay, so sometimes there is a possibility that some particles may skip from there. Okay, so now what are you going to do is you are going to go into your preliminary treatment. Sorry, primary treatment. Okay, and now primary treatment, what did I say, guys? This is nothing but it is going to be your physical treatment. Now, what happens in this case? So, guys, whatever water that we have, taken, so from here, we are going to take all this water, whatever that is there. Okay, now what happens is you are going to say that this water still has some suspended coarse materials. Okay, everything is not going to be removed in your this thing. Okay, in your the preliminary process. So you will see that there are still going to be these larger particles still floating. Okay, these coarse particles, guys, we need to remove them. Okay, and here, what is this going to happen? See, I told you this primary treatment, this is physical treatment. This is going to take place in the case of, this is going to happen in your sedimentation tanks. And now in sedimentation tank, as the name suggests, guys, what is going to happen? Either you call it sedimentation tank or the other name can be your settling tank. Okay, both the names are accepted. Settling tank or sedimentation tank. But now because of gravity, now what is going to happen? You have let this thing as it is. So slowly, all these coarse materials, guys, they are going to start coming down. They are going to start settling down. And slowly you will see that, you are going to get a blackish layer out over here, guys, at the bottom. Okay, now this blackish layer, it can be easily removed by your normal processes. And now this is nothing but you are filtering your entire sewage water. So that way. So this is what is going to happen. Now, when we are going to see, guys, when we do this treatment, we are going to see that around there is 50 to 70% reduction in the this coarse material or this solid material. Okay, so 50 to 70% reduction in your coarse particles or coarse. Okay. And now, guys, another thing that you're going to see is just like we have 50 to 70%, you are going to have 30 to 40% reduction in the number of microorganisms as well. Okay, so reduction in microorganisms. But now, which microorganisms are we talking about? Guys, we are going to get especially in your stomach and basically especially in your intestines, we are going to encounter E. coli. Okay, Escherichia coli. Or if you want, you can just write down these as your coliforms. Okay, coliforms meaning those who are going to come from your colon. So we call them as coliforms. Okay, but now see guys, I told you, see all these coarse materials, they are going to settle down out over here and they are going to give you this black layer. What is this black layer? Guys, this is nothing but we call this thing as sludge. Because we get it in the primary treatment, guys, this is going to be your primary sludge. And guys, if we are going to have primary sludge, guys, what can be done with this primary sludge? This can be used for agriculture. This can be used as a fertilizer because sludge can always be used as a fertilizer. Always remember this thing, guys. Okay, so that's it. That is what is going to happen in your primary treatment. Okay, everybody all right with this thing? Guys, understood until this point? Quickly, just say yes or no. Okay, okay, good. Okay, but now guys, see, there is still a little bit of problem. Okay, see, all these, like, these were like, big chunks of your fecal matter. Okay, there were like, solid fecal matter particles and all, they got all settled now. But see, you may have always noticed that, when you defecate, okay, let's say you're using the English toilet, that is the commode. So whenever you defecate, some amount of your fecal matter dissolves into the water. Okay, it is not going to remain like, a solid lock kind of a thing always. That let's say you are having loose motions. Okay, you are having watery loose motions. You have taken a shit. Then what? Your entire fecal matter gets just dissolved into that water. So how are you going to remove that water? That is the main thing. That is an important thing. Otherwise, okay, चलो solid particle तो हम लोग निकल रहे हैं. But if you are having dissolved substances, then वो पानी वैसे ही चला जाएगा आगे. Ultimately, you are putting it into the water or the water reservoir or your stream. Then once again, it is going to be contaminated water. So I need to remove that organic material, guys. And the next treatment, that is your secondary treatment, that is for removal of your organic matter. Okay, but now guys, in this thing you are going to have two steps that are going to be present. Okay, so once again guys, I want your entire attention out over here. Okay, so whatever water that was filtered from over here, so we're going to take this water, the filtered water, and now we are going to put it into your next tank. Okay, so what is this next tank, guys? This is nothing but it is going to be your digester. Okay, it is basically going to be some kind of your digester. So let's try to see what happens in this thing. Okay, so this digester, whatever we are talking about, guys, we call this thing as your aeration tank. Okay, so let me just first put down this thing. This is going to be your secondary treatment. Okay, so now you are going to see that all this filtered this thing sewage, okay, the filtered sewage from the primary treatment, all of that it is now going to be sent out over here. Okay, this is now going to be sent out over here. Okay, but now and let me just write this thing down. So this is nothing but your filtered filtered sewage. Okay, but now this filtered sewage it has dissolved organic matter. Okay, and our aim in this thing is to remove this organic matter. But how will we remove this organic matter, guys? So what is going to happen in this case is we are going to make use of this aeration tank. So this tank that we are talking about, this is, I told you, this is kind of a digester. So this is your aeration tank. And for this aeration tank, what are we going to do, guys? We are going to have a mixer like thing. Okay, so this mixer like thing, it has these paddles. Okay, and this is going to rotate. So let me just show you. Okay, so let's imagine this is like a slow mixer. Right, and this paddles basically they are now going to just keep on rotating slowly, 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 slowly. And at the same time, guys, what are you going to do? You are going to pump in oxygen. So from this dandi that is out over there, we have a sparger. Okay, so वो सब जाने दो ये fermentation का studies है. So you are going to add oxygen into this. Okay, so you are adding oxygen, you are pumping oxygen, and plus you are mixing this entire sewage. Guys, the moment you are going to have this kind of a situation, all these aerobic bacteria, whatever that were there, these E. coli, whatever that was there, aren't they going to get a chance to increase in number? Because see, the conditions are like favorable. You are having organic matter present inside, which is like food for your bacteria. There are bacteria, plus there is going to be warmth, plus you are adding oxygen, plus there is going to be huge amounts of your food material. So all these situations is going to be like tremendously useful for this bacteria to thrive. Immediately in this case, all these bacteria they start multiplying. Okay, so okay, so let me just put this thing down. Okay, so in this aeration tank, guys, okay, one minute, secondary treatment, I will just say, okay, this is going to be your biochemical or biological or your biochemical process. Okay, so now in this aeration tank, you are going to pump oxygen, and second thing, you are going to mix that entire sewage water. They mix the sewage. Now what happens? Because of this thing, because whatever we are doing, immediately bacteria and fungi they thrive. Okay, bacteria and fungi they thrive. And now what happens, guys? You are going to see immediately this is going to result into something that is called as flock formation. Very, very important term, guys. Okay, this is going to result into flock formation. Now, what exactly is flock formation, guys? You are going to see that these fungi, whatever that were there, they start growing like this, like this, like this. But guys, when fungi grow, fungi are going to have mycelium, right? So this is whatever I'm showing. This is going to be your mycelium. Okay, this is going to be mycelium of your fungi. Okay, and what you're going to see that in this mycelium of fungi, you are now going to have the bacteria. They are going to be entangled inside, meaning they are like, just stuck out over there. So these are going to be bacteria, and together we call them as flocks. Okay, so bacteria entangled in the mycelium of the fungus, that is known as your flock formation or flock. Okay, so bacteria entangled in the mycelium. Okay, fine. Now what happens, guys? You are going to see that because these bacteria are present, okay, so now you will see a situation. Now see slowly, slowly, guys, how it is going to happen. Slowly, slowly, you are going to see that, oh my God, these flocks, they start coming up. Like, these are, just imagine these as like, small, hair gunta, if females generally, when you comb your hair suddenly you get hair, what are you going to do? You are going to roll it into a ball like this, hairy ball, hairy mass kind of thing. This is exactly like that. Just imagine it to be something of this sort. Okay, so you are going to see that, you are going to have flock formation taking place. Okay, so let me just write this them down. These are nothing but your flocks. Okay, but now, guys, what is the job of the flocks? See, these flocks, they are like thriving, they are surviving, they are like enjoying out over there. So initially what they do is, guys, these flocks, they start eating or they start breaking down your organic matter. They start breaking down organic matter. But now, guys, what happens? Okay, let's understand this thing. Okay, whenever you start breaking down organic matter, guys, you are always going to require more oxygen. Okay, always remember. And because of this, while we are requiring more oxygen, see, this is the real reason we are like, pumping this oxygen into this medium. Okay, so that these flocks still survive, they are going to still thrive and they continue breaking down your organic matter. Okay, so now let me just give you an idea. Okay, so guys, at the start of secondary treatment, there was large amounts of contamination. Okay, there was large amounts of contamination in that sewage water. So that contamination, we actually, we measure it in terms of BOD. So we call it as biological oxygen demand. Okay, so now guys, see, in the start, this was highly polluted. Okay, so because see, when we started out over here, all that organic matter laced with sewage, it came out over here. We had to break it down. Okay, now guys, please understand this thing. At the start of this treatment, the biological oxygen demand, the need for oxygen was very high because there was more amounts of organic matter. So we say that BOD is very high, and whenever BOD of anything is very high, it indicates it is highly contaminated. It indicates that whatever that water body is, it is going to be highly contaminated. Okay, but now as the process takes place and towards the end, guys, towards the end, these flocks and all, they are going to break down your organic matter, right? So after that organic matter has broken down, guys, are you still going to have excessive oxygen demand for this thing? No, the demand for oxygen is going to decrease because there is less material to be decomposed. There's less material to be broken down. So towards the end of your secondary treatment, guys, now you're going to see that the BOD decreases. And if the BOD decreases, guys, then what does this mean? That now it is going to be less contaminated or the contamination has gone away. It depends upon what levels of low BOD you are looking at. Okay, less contamination. Okay, everybody all right with this thing? Understood this part, guys? Always remember in your exams, they will ask you this thing. What is BOD and what is high BOD and low BOD responsible for? Okay, so now what happens, guys? Now we are going to bring about this thing. Okay, we are going to bring about this entire secondary treatment, and we are going to see that, oh, slowly, while we do this thing, the BOD of this water decreases. Okay, now the biological oxygen demand of this water as it decreases, now we know, अरे चलो इसके अंदर का सब organic matter has been utilized, it has been broken down. Once that thing is done, guys, now what are we going to do? We are going to take this water and now we are going to send it to the next tank. Okay, now what is this next tank, guys? The next tank is once again your sedimentation tank or it is going to be your settling tank. Okay, now this is a part of your secondary treatment only, guys. Okay, we have not reached into your tertiary treatment. So all this water, whatever that is there, this is now going to be sent out over here. Okay, fine. Chalo, so this is out over here. Okay, now what is going to happen, guys? Okay, so now you are going to send it into your next step. Okay, you are going to send it into the settling tank. Now, guys, there is a little bit of discrepancy. Okay, now what is this discrepancy, guys? Like in your textbooks, okay, if you look into your textbook, this settling tank in this year's edition, they have put this thing in your tertiary treatment. Okay. However, according to your last year's textbook or even previous year's textbook, this was a part of your secondary treatment, guys. Okay. In fact, even if you open up your this thing, like textbook and you look at it, okay, you will see that it is a part of your secondary treatment only. But by mistake, they have like added it into the tertiary treatment. So I will stick to the normal one, guys. Okay, we will stick to how it is really going to be like. Okay, so it is going to be a part of secondary treatment. Okay, so this is also your secondary treatment. Okay, so keep this thing in mind. Okay, so now what happens is, guys, whenever you are going to transfer this water, along with this, you are going to see that, oh, some of the flocks, they are also going to come out over here because it is not going to be possible that, all the flocks are going to be removed. Okay, so now what happens? Oh, we have got your flocks out over here. Okay, now as what are we going to see? This is nothing but it is going to be your settling tank, or we said this is going to be your sedimentation tank, right? Settling or sedimentation tank. So in this case, now what is going to happen? All these things, once again, they are going to come down and now they are going to once again settle down out over here. Okay, so some of them you are going to settle it down. So once again, what is this going to give you, guys? This is going to give you your sludge, right? And this sludge, whatever that is there, now we term this thing as activated sludge. This is going to be activated sludge. Guys, once again, what can this sludge be used as? This can be directly used as your fertilizer. Okay, so now what happens? See, some of the flocks they have settled at the bottom. Okay, so let me just put it down out over here. So in this treatment, you are going to see that the flocks settle at the bottom and they are going to give rise to activated sludge. Okay, but now guys, now what happens is, see, I want more flock formation out over here in your secondary treatment, in your aeration tank. Because if the flock formation happens properly, guys, then what will happen? The organic matter will be broken down really easily. It will be more faster and the process will take less time. So in order to stimulate more flock formation, what are we going to do? We are going to take these some of the flocks and we are going to put it back into your aeration tank. This is known as your seeding process. So I will make a note out over here. Some flocks I transferred back into your aeration tank. Again, this kind of a process, this is known as seeding process. Okay, what is this seeding process going to do? It is going to increase the rate of your flock formation. I'm sure you guys must have seen seeding in chemistry. Like, if you have a saturated solution, salt solution, and you want to crystallize salt out over from there, what are you going to do? You are going to add a little bit more of your salt. Okay, and that salt crystal, what are they going to do? They are going to initiate that entire seeding process and immediately you are going to see that salt starts crystallizing out from that saturated solution. This is what is going to happen. Everybody okay with this, guys? Understood this part? Fine. Okay, any doubts in this thing, guys? So this is where our secondary treatment is going to end, guys. Okay, quickly let me know if everybody is okay with this. Okay, okay, fine. Okay, but now let's try to move ahead, guys, and let's try to see the next thing that is the last treatment. Okay, now in this last treatment, guys, see, until now, what were there? So we had said that, oh, we are going to have your aerobic bacteria. This is going to contain aerobic bacteria. This flocks and all, they are nothing but your, fungi and bacteria. They are going to be there. But can I take this sewage water like this and I can put it into your water bodies? No, because then all the bacteria are going to enter, all the fungi are going to enter into your water bodies. I don't want that thing to happen. I want to stop it from happening. So now what are we going to do, guys? After this secondary treatment, okay, so let me just once again, if you cannot see this thing, so this was our secondary treatment. Now, guys, after this secondary treatment, now whatever this water that is there, the filtered water, okay, that is now going to be put into your anaerobic digester. Okay, so now let me just go and show you another last one. Okay, now this last chamber, guys. Okay, this last chamber, this is our anaerobic digester. Now, guys, what is the job of this anaerobic digester? Okay, let's figure this thing out. Okay, so this is our tertiary treatment. Okay, so now, guys, this is going to be anaerobic digester. Okay, now in this, what we are going to do, guys, we are going to use anaerobic bacteria. And likewise, see, we had earlier said that, we are going to have anaerobic bacteria like Clostridium, Pseudomonas, right? The same bacteria we are going to add out over here. So you are going to have anaerobic bacteria, and that is nothing but your Clostridium. We are going to have Pseudomonas. Now, guys, the moment you are going to add them into this, what is going to happen? These are the ones they start eating, okay? They start breaking down all your aerobic bacteria and fungi. Okay, so immediately this is going to break down aerobic bacteria and fungi. So now all that thing that was there in the secondary treatment, they are going to be killed. That flocks, whatever that was there, it is going to be killed. And now what? Once that has been killed, at the same time, they are also going to work on like whatever left or organic matter that may be there. So what are they going to give out, guys? They are going to give out gases like CH4, or maybe you are going to have CO2, or maybe you are going to have H2, or maybe you are going to have your H2S, or maybe you are going to have a little bit of H2, or you are going to have some trace gases. Even these are going to be released during this entire process. And now, once this entire tertiary treatment is done, then what are you going to do? You are going to take these, and now the water is let out into your water bodies. So now the effluent is released in water bodies. But now the next question that may come to your mind, but sir, we have treated all the aerobic bacteria. Aerobic bacteria were killed, but now this water also had your anaerobic bacteria, right? But now if you're releasing that into the water body, that means you are adding anaerobic bacteria into the stream. No, 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 guys. Now what is going to happen? Understand this thing. Okay, so you are going to see that whenever we take this and the process from this anaerobic digester, and when we release this anaerobic bacteria cannot sustain oxygen in the environment. Then what will happen? The moment they come in contact with oxygen, kacha, 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 they are going to die. And the moment they are going to die, guys, now what is going to happen? Immediately you're going to see that the water is going to be clean. But then, along with this, guys, to be more, okay, you are going to see, you have to be a little bit more careful. So, what are you going to do, guys? You are going to bring about chlorination. So, you are going to add huge amounts of chlorine into that water body as well, like the effluent. So, you are going to bring about chlorination. And now, later on, whatever that effluent is there, you are now going to release it into your water bodies. Okay with this thing? Guys, everybody all right with this? So this is this entire process of your sewage treatment plant. Really, really huge stuff. But then, अरे ये ज़्यादा दूर हो गया. एक second. Okay, but this is how that entire process works out to be. Okay, just a moment, guys. मैं थोड़ा सा इसको नजदीक ले लेता हूँ. Okay, so this is it, guys. Okay, so once again, my suggestion to you is quickly just go through this. I'm not sure whether you'll be like properly seeing this. Okay, but thoda sa just try. Okay, and guys, Do this in this form only, because you know why? If you read it from the textbook, okay, guys, it is going to be really, really crazy. Okay, you are going to go a little bit crazy, and then you may forget. Okay, so that is why the easiest way is this way. One minute. Okay. Okay, everybody all right with this, guys? Everyone? Okay, just go through this, take your time. Okay, guys, what I will do is I'll give you a quick 5 minute break. Okay, if anybody wishes to go to the loo, use the washroom, or have some water, quickly, just in 5 minutes, quickly go. Then we are going to see the next part, like, how we can use your microorganisms in your biocontrol. Okay, biocontrol, biofertilizers. So we have to see that as the next part. Okay, chalo. So let's move ahead, guys. Is everybody done? Fine. Okay, is everybody back? Give me a yes or no. Quickly, 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 guys. Yeah. Okay. Sure. So, guys, now after your sewage treatment, guys, remember this question is very, very important. Okay. So, don't miss this thing under any circumstances. Okay. Sure. So, now the next thing that we're going to talk about is, guys, now how are we going to use your microorganisms in your biological control? Meaning, how can you use it to control your pest? How can you use it to control your moths or maybe flies, grasshoppers, caterpillars? How are we going to do that? So the next thing, the next part of the chapter, slowly, slowly, we are like reaching the end of the chapter, guys. Okay. So, okay. So we are going to have use of microorganisms as biocontrol agents. Okay, use of microbes as biocontrol agents. So, guys, there are two things out over here. Microbes can be used at first. They can be used as your biocontrol. Basically, they can be used in biocontrol, or they can be used as your biopesticides. I will just explain you what is the difference between the two. Okay, so whenever we talk about your biocontrol, guys, it is nothing but the use of biological organisms. Okay, it is the use of biological organisms for controlling, okay, for controlling your plant disease or your pests, for controlling plant diseases or pests. Okay, so this is biocontrol. So whenever you incorporate any bacteria to maybe prevent some disease from happening, then that is biocontrol. But then what is biopesticide? Okay, there is a slight difference between the two. Okay, so biopesticide is nothing but use of any biological agent. Okay, so biological agent or your organism. Okay, it is to kill. Okay, this is generally done to kill or check the proliferation of. To kill or check the proliferation of disease-causing agents. Okay, to check meaning to kill or to check meaning to control the proliferation of disease causing agents. Right? So basically, both of them they are going to be on the same lines. However, there is a very thin difference between the two. Okay, but now, guys, why are we going to go for your microorganisms to control your pests or to control your diseases? See, we have been traditionally using chemicals. Okay, so chemical pesticides, chemical fertilizers. Hey, from that, why are we moving into biopesticides, biofertilizers? What is the reason behind it? So the problem with chemical fertilizers or problems, okay, the problem with chemical pesticides, okay, that is the ones that are normally used. Like, what is the problem with them? Let's try to talk about. Okay, so problem with chemical pesticides, that is the normal ones. First of all, these are going to be harmful. Okay, they are going to be harmful and toxic to your humans as well. Okay, so if you have this pesticide-laden fruit and you haven't really washed it, then what is going to happen is that pesticide is going to enter into your body. And once it enters into your body, guys, it is not going to be eliminated from your body. It is going to stay in your body and it is going to start accumulating. That is the problem. Second thing, guys, whenever you spray these chemical pesticides, 50% is going to fall onto the crop. The rest 50%, it is going to fall onto the ground. And when it falls onto the ground, guys, what is going to happen? It seeps into the ground. Okay, now as it seeps into the ground, now what is going to happen? It is going to reach the lower layers of the earth, and from the lower layers, it is going to enter into your underground water. And that is why it contaminates underground water. Okay, so this is the second thing, guys, that is going to happen. Okay, but now the third and the worst thing, okay, the third and the worst thing is, guys, see, these chemical pesticides, they are really, really potent, meaning they are really, really effective. But now, do they really understand which is a good bacteria or which is a bad bacteria? Now, in your soil, there are going to be so many good bacteria, right? But then this does not, make that discrimination. It does not make a distinction who is good, who is bad. And this is going to kill all the organisms, kills all the organisms, irrespective whether it is good or bad, indiscriminately. It kills all the organisms indiscriminately, and which is going to be really, really bad. So we don't want this kind of a situation. So that is why we are going to go for incorporation of a biological agent to control these pests. And how is it going to be achieved, guys? This can be achieved in two ways. Okay. So the first example, how we can go for this is by making use of your Bacillus thuringiensis spores. Okay. So let's try to see, guys. So guys, this Bacillus thuringiensis. Okay, now what is this? So, a second, I'll just put it down. Thuringiensis. Okay, so this guy, Bacillus thuringiensis, guys, this is a bacteria. Okay, and this bacteria, you may have heard about it. See, guys, have you ever heard of BT cotton? BT cotton, BT brinjal, BT bhendi, right? That BT is nothing but this Bacillus thuringiensis. Okay, so now, guys, what happens out over here when we talk about this Bacillus thuringiensis? Why is it really important? So, guys, this Bacillus, it actually produces spores. Okay, so that is natural for this thing. So, what we do is we take these spores and we are going to mix them in water. And now we take this spore and water mixture and we are going to spray over the field. Okay, fine, spray over field. Now what happens, guys? Okay, now slowly you're going to see that these spores, they are going to be, lodged onto the leaves, they are going to be lodged onto the stem and all. So what happens is when the prey eats it, okay, meaning when the this thing, the pest basically, when the prey eats this thing, when the prey eats it, guys, what is going to happen is the spores are going to enter into the digestive tract. Okay. Now, as the spores enter digestive tract, guys, now what happens in the alkaline medium of the this thing? Okay, in the alkaline medium of the intestines, the spores are activated. And now, guys, the moment the spores are activated, what happens is they are going to produce something which is called as Bt toxin. Okay, so one minute. They are going to produce something that is known as Bt toxin. But now, what is this toxin going to do, guys? The moment it is produced, it is going to bring about inflammation of your intestines. Okay, so inflammation of intestines, and then this is going to result into your vomiting, discomfort, excessive pain, and ultimately what? This is going to result into the death of the organism this way. So it results into the death of your prey, like whether it is your grasshopper or whether it is going to be any kind of your moth, whoever eats it, immediately this is going to die. So, guys, isn't this going to be like a wonderful thing? Like, you are using a biological organism against a biological entity. So this is what is your biocontrol, guys. Okay, but then you may say, sir, but what if? What if this is going to enter into humans? Then what? So fret not, guys. Don't worry. The reason is because humans have an enzyme which can break down this, this toxin. So this is not going to have any effect onto the human body. So this is going to be completely safe. So guys, this is going to be a win-win kind of a situation for both of us. And that is why slowly you are seeing that, oh, now we are moving from your chemical pesticides to your biological pesticides or biocontrol elements. So this is going to be one, guys. Okay. But then let's try to see the second thing. Okay. The second way we are going to achieve the same kind of thing, that is going to be with the help of trichoderma species. Okay, so this trichoderma, okay, especially we have trichoderma coniG that produces your cellulase. Okay, so we have this trichoderma. Okay, so this is basically your free living fungus, basically found in your soil, free living fungi in your soil. Now, guys, what is the most important thing is, right? This is going to be against any kind of your soil, fungal pathogen. So this works against, I will just mention, against fungal pathogens that infect the root. Okay, so or I will say it works against soil-borne fungal pathogens. Okay, now what is going to happen is this fungus, that is this trichoderma, how does it protect the plant? Okay, so what it does is it produces different kinds of compounds. Like what? We are going to have glyovirin. Okay, so we have something called as glyovirin, something that is also called as your glyotoxin. Okay, so we have glyovirin, glyotoxin, and something that is your viridin. Okay, so viridin. Guys, these are certain compounds that it is going to produce. And now, what is this going to do? All these things, they are going to inhibit your soil-borne pathogens. That's it. That is what we need to know with regards to your trichoderma, guys. Everybody okay with this thing? Fine, no problem. Okay, sir, you have written by mistake prey instead of pest. The prey is because it is going to eat it. Na? That is why it is going to be the prey only. Okay, so basically this ultimately this is going to die. So you can write it as prey or it can write it as pest. It is the same thing. No, no, here the idea is I am going to kill it. So that is why it is my prey. So that way. Okay, so guys, this is what is going to happen with regards to your this thing, that is your biocontrol. Okay, but now the next thing that we are going to come up with is your this thing, guys. Okay, that is going to be your microbes in your bio pesticides. Okay, so let's try to see how is it going to be in your biopesticides and who are we going to act. Okay, guys, this is very important, but then you will have to remember this thing. Okay, so biopesticides, who is going to work on whom? Okay, I will give you a way to remember it, but still, okay, I will just show you how what really happens out over here. Okay, so guys, what I'm going to do is I'm going to take this, I have taken this picture directly from your textbook, okay? Because there are quite a few names out over here, but I will give you a way how to remember it. Okay, so guys, in your biopesticides, who are we going to use? We are going to have different kinds of your this thing. Okay, so we are going to make use of bacteria. Okay, so we have bacteria also. Okay, we are going to have your fungi, bacteria, fungi. Then we are going to have your protozoans, And finally, we are going to have even your viruses, guys. Okay, even viruses, all of them can be used. Okay, but now the question is, if we are talking about your bacteria, who are we talking about? So the most important one, it is going to be your Bacillus thuringiensis. This is the most important one, guys. Okay, Bacillus thuringiensis, Bacillus papillae, and Bacillus lentimorbus. Okay, all of them, they are going to be like working against what? They are going to be working against your caterpillars, cabbage worms, adult beetles. And along with this, what they have missed is you are going to see that they are also going to work against the larvae of Aedes mosquito. This is what they have forgotten. Larvae of Aedes mosquito. Okay. So first thing is, guys, how are you going to remember this? Now, I like to remember this as say cab. Okay. So one is your caterpillars. Second is your cabbage. A is for your adult beetles, and then B is for beetles, and A is for your larvae of your Aedes mosquito. So I like to remember it in this way. Okay, but now the next thing we are going to have is your fungus, and out of this fungus, this one, the first one is the most important, Beauveria bassiana. Bavaria bassiana, or it is going to be the second one, Entomopthora. Then we Entomopthora pallidarosium, and then we have Zoophthora radicans. Okay, these names, guys, you need to remember. They are a little difficult to pronounce, but you need to remember. I remember this as BZ. Okay, so fungi. I am going to remember this as BZ, ma'am. Okay, I will busy ma'am W. Okay, this is generally how I remember this thing. Okay, you will have to come up with your own this thing. So busy ma'am W. Okay, so M is for what? M is for your mealybugs. A is for aphids. Okay, aphids meaning you are going to have aphids croci, aphids unguiculata, both. And then next M is for mites and W for your white flies. So ma'am W. Okay, it doesn't make sense, but it will help you to remember it. Okay, next one we have your Nosema locustae. Okay, so basically this is going to be your the one who's going to attack against your locust. Okay, now this is locustae. It is not lacustae. It is locustae. Okay, but now what is this going to work against? This is going to work against grasshoppers, caterpillars, and crickets. Okay, there is one more out over here that is going to be your corn borers. Corn borers. How do I like to remember this? I like to remember this as GC3. Okay, or GCQ. So grasshoppers, caterpillars, cricket, and corn borers. And then the last one, we are going to have your viruses. In viruses, guys, we are going to have NPV, nucleopolyhedrovirus. And you know what? There are actually 125 different types of your NPV. Okay, 125 different types. And then the second one we are going to have is your granulovirus. So NPV and granulovirus. What are they going to be against? They are going to be against your caterpillars. Once again, the same thing, gypsy moths, ants, wasps, and beetle. Now, out of this, guys, you need to remember gypsy moths because in your exams, they will ask you this thing, guys. Okay, gypsy moths. Who is going to work against gypsy moths? How do I like to remember this thing is, okay, this is going to be GC bow. Okay, I like to remember this as GC bow. So gypsy moth, we have your gypsy moth, caterpillars, then we have beetles, we have ants and wasps. Okay. Now, guys, I know this is going to be a little confusing for the first time, but as you go ahead, okay, as you move ahead and you keep on reading this thing, reading this thing, reading this thing, slowly, it is going to fit into your brains. Okay. Remember this thing. So you just remember this thing as when we talk about bacillus, you are talking about cab. Okay. So just remember this thing. If you're talking about fungi, you remember BZ MAM W. BZ MAM W. If you're talking about Nosema locustae, okay, you are talking about GC3. Okay, but here that G is your grasshopper. This is not your gypsy moths. Okay, And finally, you have your NPV or your granulovirus, then this is going to be your GC bow. Okay, just remember, try remembering this way, guys, and you will see that, it makes things really, really easy. Okay, guys, just have a look at this. Just see if you have any doubts, if you wish to ask anything. Okay, everybody all right with this, guys? Fine. Okay, now just like how we have used your, microbes as your biopesticides, we also use them as your bioherbicides. Okay, and in the bioherbicides, we have two. If you're using your bacteria, then it is going to be your bacterial herbicides. And if you're using your fungi, then it becomes your mycoherbicides. Okay, so but then we will talk about those, but we'll talk about them in the next class. So for the time being, guys, we'll stop over here. Next class, we're going to finish this thing off. And what I will do is I'll see if we can start off with a new chapter as well in the next class. Okay, difference of pesticides and herbicides. See, when we talk about pesticides, they are going to be against some pests. Pest is like, it can be your crickets, moths, butterflies, it can be caterpillars. Okay, so these are pests. But when we talk about herbicide, they are going to be used against some, so it can be your bacteria, which can be used against plants. So herbicide is against a plant, pesticide is against a pest. So that way. Okay, so it can be bacteria used against a plant or fungi used against a plant. That way. Sir, in sewage treatment, we can add Ganga and Yamuna action plan in theory exam. Yes, definitely. You can add that as well. Okay, any extra information you wish to put, you can always put it out over there. Okay, sure. So guys, once again, we'll stop over here for the day and I will see you in the next class. Till then, take care. Bye bye, everybody. Bye bye. Bye. Bye.